We first left off at the pancreas, so let's go ahead and dive into that. First, let's discuss the anatomy of the pancreas. Like we saw in lab, you would have to lift up the stomach in order to see this pancreas here. You'll also see that the head of the pancreas is cradled by the duodenum over here, which is the first part of the small intestines. And then its body spans across what we would have vertebrae back in here, and the tail will point to the spleen. Now, the pancreas is a really interesting organ because it has both endocrine and exocrine functions. We'll see that in the histology slides to the right here, we have these pancreatic acinus for singular and acinar cells for plural that are going to be contributing to the exocrine function. So here we would have secretions of some pancreatic juices in order to assist with digestion. So that's its exocrine function, and it makes up the vast majority of the pancreas. Now we do have these other little, I'm going to call them islands, that are called pancreatic islets of Langerhans, and these are going to contain our endocrine cells. Within this group, we will have some alpha cells here that are responsible for secreting glucagon, and our beta cells in purple here that secrete insulin. So now let's talk about those pancreatic hormones. They're going to work together in order to maintain our blood glucose, and the normal range for that is going to be 70 to 110 milligrams of glucose per deciliter. And if we have high levels of this glucose, then our blood vessels and kidneys become damaged. And if we have too low of levels, then we could get lethargy, mental and physical impairment, and even death. So how are we going to lower glucose when we have high concentration in our blood over here? What happens is that our pancreas is going to secrete its insulin, which will move into our bloodstream, and then insulin will work to take that glucose and basically move it into cells. So we are going to start moving it into the liver tissue so that we can increase glycogenesis, which is basically taking the glucose molecules and building it into bigger molecules called glycogen. And we're going to decrease the glycogenolysis, meaning the breaking down of that big molecule glycogen, so that we get smaller glucose molecules, right? We don't want to do that. We want to store that glucose rather than break these big molecules apart and have little glucoses running around. And then we also are going to decrease gluconeogenesis. And this is a process in which we're going to use non-carbohydrate substrates. And this could be anything like from lactate, amino acid, and glycerol, and transform it into glucose. So we don't want more glucose. We want to decrease it. So that's why that process is decreased. And then the other thing is we can move that glucose into our adipose connective tissues by increasing lipogenesis. So we're adding the fat in here, and also decreasing lipolysis, meaning we're breaking this fat apart in order to get some of those molecular components out. Like, for instance, we can take out glycerol out of there and go through the process of gluconeogenesis to create glucose if we need it. We can also move that into our other cells of our body, especially our muscle cells, so that we can use things like amino acid in order to stimulate protein anabolism. And then here are our other cells, most of our cells, so that we can use that glucose to create things like ATP in our cells to power them up. So I know we went into detail there, but let's just review what happened. We ate some food, let's say we got some glucose in our blood. So our pancreas, specifically our beta cells here, got stimulated, released insulin, which traveled through the blood. And then insulin went looking for receptors on its cells. Now, just a quick note that some of the cells don't require insulin to take in glucose, and that would include neurons, our kidney cells, hepatocytes involved in our liver tissue, and also red blood cells. But let's say the insulin travels, finds a receptor on an adipose cell, and will bind, and then that opens a channel here to allow the glucose to move in. So as insulin is moving all of these glucose channels to allow glucose to move into our cells, the glucose concentration in our blood is decreasing. So now we are going to have this negative feedback loop where we get a signal to our pancreas beta cells telling it to decrease its concentration or secretion of insulin because our blood glucose level is within a normal range. Now, what happens if we need to raise our blood glucose levels because they are too low? So if you, let's say, are not eating and we don't have any glucose, not any, but we have a decreased glucose concentration within our blood, then in our pancreas, our alpha cells are called upon, and that glucagon is secreted into concentration in our blood. And here glucagon is going to act through those membrane receptors and second messengers to cause the body cells to release any stored nutrients into our blood. So we can travel over to our liver, and the hepatocytes, liver cells here, will release glucose and put that back into the blood. We could do this by increasing glycogenolysis. Remember, that's breaking apart glycogen to get those glucose cells. And then we can go through gluconeogenesis, where we are going to use non-carbohydrate substrates in order to make glucose and release it there. And then we want to decrease glycogenesis in order to use any of those glucose molecules that would be used for this process to instead release the glucose into our blood. And then we can use our adipose cells to release fatty acids and glycerol. So that is going to increase lipolysis to break that apart and get those components, and decrease lipogenesis, so making these adipose cells store that glucose. We don't want to do that. And so, like we said, we can use these components in order to get glucose into our blood. So glycogen can be given by paramedics to unconscious individuals with low blood sugar. Whether it's through that process or your body's natural way of releasing glucose into your blood, we are going to get a negative feedback loop once again, and our glucagon levels are now going to become inhibited because we realize that our blood glucose is within a normal range and we don't need glucagon as much to be secreted to raise that blood glucose anymore.